people. Now we'll begin our session on structural and cellular organization. One quick note, is Mary Miller here today? Okay, never mind then. So without any further ado, Alison Walters from NIH will tell us about nuclear morphology.
I hope you can see me behind here. Okay. Yeah, I want to say thanks for the chance to talk about my work today. I'm a postdoc in Ornico and Fixit's lab, and today I'm going to tell you about my work on the role of Cdc5 in regulating both cell growth during a mitotic arrest and nuclear morphology. So it's long been known that nuclei from any given cell type have a very specific shape. And here I'm showing you some very nice, beautifully round nuclei from a C. elegans embryo. It's also been recognized for a long time that the volume of the nucleus to the cell volume is a ratio that's also very constant. But how the nucleus stays in shape and how this ratio is maintained is not something that we understand very well. What we do know, however, is that the nuclear morphology can be altered in both aging and in several disease states. So here I'm showing you these nice smooth nuclei from a 9-year-old and these somewhat wrinkly ones from a 96-year-old. In cancer, abnormal nuclear morphology and size is used as a diagnostic tool. And here you can see from these lung cancer cells, these nuclei are enlarged and they have invaginations in the nuclear envelope. And even though it's used as a diagnostic tool, people don't really understand the cause-consequence relationship in terms of the disease. And so in our lab, we used yeast as a model to try and study how the nucleus normally stays in shape, how it regulates its size, and it's our hope that if we can try and understand this a little better, we might have a better understanding of what's going on in these disease states. So a previous screen in our lab led to the interesting observation that in yeast, the nuclear envelope continues to expand during a mitotic delay. So here I'm showing you a wild-type cell progressing through mitosis. You can see here at this time point with the nucleolus, the nucleoplasm shown in green and the nucleolus in red, and I'll use those colors throughout my talk. You can see at this particular time point the expansion of the nucleus, its elongation, this formation of a dumbbell shape, and then the formation of the two daughters. Now, if you take a wild-type cell and arrest it in mitosis, and here we've done that using nicodazole, which is a drug that depolymerizes the microtubules and arrests the cells at metaphase. And what you can see here, with approximately similar timing to what we see in the cycling cell, is that the nuclear envelope continues to expand, even though the cell cycle has stopped. And this expansion occurs specifically at the site of the nucleolus, and we call this a flare. And so this observation led to the idea that perhaps the nucleolus is acting as a membrane sink during the mitotic delay. So here in this cartoon, I'm showing that if a cell is progressing through mitosis as normal, nuclear envelope expands. This allows anaphase to be completed, the segregation of the chromosomes. If the cell's delaying in mitosis, nuclear envelope expansion still occurs, but the additional membrane is sequestered at a site adjacent to the nucleolus, and it forms this flare. So we hypothesized that perhaps the sequestration of this membrane at this particular site allows the maintenance of the nuclear cell volume ratio in the face of a big expansion of the nucleolus, but, bless you, but not much expansion of the cell, or in the face of no nuclear division. And so you can see that if you put all this extra membrane in an extension, you're going to increase the volume of the nucleus a lot less than if you expanded the nucleus isometrically. And so this all leads to some very interesting questions. Firstly, how is nuclear envelope expansion achieved in mitosis? What are the mechanisms that control this process? And really interestingly, what restricts this expansion to the nucleolar region? So I carried out a screen. This is where I came in. I entered the lab and I carried out a screen to try and identify mutants where, when the cell arrests in mitosis, no flare is formed. And I screened lots of TS mutants, and the one I'm going to talk about today, the hit that I found, is a mutation in Cdc5. And so here I'm showing you at 23 degrees wild-type cells, and this Cdc5NF, which stands for no flare mutant. And you can see they form this flare structure. At 34 degrees, which is the non-permissive temperature for the Cdc5 mutant, you can see that under these conditions, wild type still forms flares, but the Cdc5 mutant, the nuclei is still round. And this is a really penetrant phenotype. Only around 5% of the cells you will see a flare. And so Cdc5 is a really well-studied kinase. It has multiple rows of mitosis. It's homologous to polo kinases 1, 3, 4 in mammals. Has an N-terminal kinase domain and C-terminal polo box domains. So the mutation I've identified is here in the kinase domain, but we have looked at other Cdc5 mutants and also a Cdc5 degron strain, and in all of them they have this no-flare phenotype. And so it's not allele-specific. So as I said, Cdc5 plays a plethora of roles in mitosis. In yeast, its essential role is in mitotic exit. Previous work, which I'm not going to have time to talk about today, but I actually talked about at the previous meeting, we don't think that Cdc5 is acting through any of these known roles in mitosis to affect nuclear morphology. So how is it affecting nuclear envelope expansion? And here there's a few models we can come up with. One is that perhaps Cdc5 positively regulates nuclear expansion. So for example, you need Cdc5 to upregulate lipid synthesis when you hit metaphase to allow expansion to occur. So in the Cdc5 mutant, expansion simply doesn't happen, so you don't make a flare. The second option is that perhaps Cdc5 regulates the addition and retention of the membrane specifically at the nucleolus site. So in the absence of it, membrane's just added all over, and that's why you stay round. And the third option, which is a somewhat less direct mechanism, is that perhaps Cdc5 normally is involved in inhibiting cell growth during mitosis, and so this makes flare formation necessary in the wild-type cell because nuclear expansion is happening faster than cell growth. So perhaps in the Cdc5 mutant, the cell continues to grow, so the nucleus can carry on growing isometrically, and it doesn't disturb the nuclear cell volume ratio. Previous work, again, I don't have time to discuss, we've discounted model 1 because we looked at Cdc5 no-flare nuclei and see that they do indeed expand during a mitotic arrest. So the next model we decided to test is model 3 here, and that is that the Cdc5 is inhibiting cell growth. And so we first wanted to ask, if we compare wild-type and Cdc5 no-flare, does the mutant grow more during a mitotic arrest than the wild-type cells? So this is actually an experiment that Orna did, so if you have any problems with it, see her. We took the wild-type and Cdc5 no-flare strains and added nicotazole and shifted them to 34 degrees, so to the non-permissive temperature. And then Orna took samples 2, 3, 4, and 5 hours after she'd added the nicotazole and imaged the cells and then measured them. And here I'm showing you time in nicotazole on the x-axis and cell volumes on the y-axis. And wild-type duplicates are in blue, and the Cdc5 no-flare is in red and orange. And you can see that while wild-type increases in size a small amount during this time period, the Cdc5 no-flare mutant increases in size considerably more. And so Cdc5 mutant is growing more than wild-type, but is this the reason why the nucleus is a different shape? And so if the cell growth phenotype is the reason for the no-flare, then if we can now inhibit the growth that we see in Cdc5, can we now restore the flare phenotype? And the way we went about trying to do that is using mutations in the SEC pathway. And so we specifically used mutations in SEC6 and SEC15, which are both components of the exocyst complex, which is required for vesicle fusion at the plasma membrane. And we combined these mutations with the Cdc5 no-flare mutation. We took these strains and we arrested them in nicotazole at 23 degrees, so this is the permissive temperature, for 3 hours. And then our time 0 point, we will have all of these strains arrested in metaphase, and they would all have flared nuclei. We then shifted them to 34 degrees and maintained the metaphase arrest. We did that for 2 hours, and then we took another sample. Now, I should say that from many previous experiments we did characterizing the Cdc5 no-flare phenotype, we know that the Cdc5 no-flare nucleus under these conditions will start flared at time 0, but when we shift it for 2 hours, it will lose the flare and become round. And so when we look at the cell growth measurements, we can see that, as we saw before, in a similar experiment, wild-type grows somewhat, and Cdc5 no-flare grows considerably more. The SEC mutants on their own don't grow, and most importantly for us, we saw that by adding the SEC mutations into the Cdc5 no-flare background, we've now inhibited its growth. And so this is exciting. What's happened to the nucleus? Well, life isn't simple, and as proof of that, when we started looking at these sec mutants, we realized they actually have a very interesting nuclear phenotype of their own. Here I'm showing you just wild-type cells that have these nice round nuclei. When you look at sec mutants, here I'm showing you sec6-4, but it's true for all of the sec mutants I've looked at so far. These are asynchronous cells shifted to 34 degrees for 2 hours. You see they have this really striking bilobe nuclear phenotype, where the nucleolus is in one lobe of the bilobe. And when you look at these cells using DAPI, so if you look at them at their DNA, they look perfectly normal because the bulk of the DNA is in this lobe here. And so they're doing something very strange. When you add an inhibitor fatty acid synthesis, you inhibit this bilobe formation, so the nuclei remain round. So perhaps what we see here is something similar to what we're seeing in mitosis, and that is that the mutation in the SEC pathway is inhibiting cell growth, but the growth of the nucleus is continuing, and it's altering its shape to try and maintain a ratio. That's just a hypothesis at this point. So this is a really, really interesting observation, and has led to somewhat of a digression in some of my work. But the experiment we were originally trying to do is to work out what's going on with the Cdc5 no-flare nuclei. And this altered nuclear shape obviously makes life a little difficult in terms of interpretation of those results, but we went ahead and looked at the results and looked to see if we can learn something from them anyway. And so this is the same experiment I showed you before, when we look at cell size and nuclear morphology before and after a 2-hour shift to 34 degrees. This is the same cell size data where I show you that Cdc5 no-flare grows, and when we add in the SEC mutation, it doesn't. So here I'm showing you the data for nuclear morphology at just the 2-hour time point. In wild type, they maintain their flares to pretty much 100%. Cdc5 no-flare, as I mentioned before, loses flares and the nuclei become round. Around 40% maintain their flares. SEC mutants mostly have extensions of the nucleolus, and this is mostly actually in the form of a bilobe. And we would predict if Cdc5 cell growth phenotype is the reason for the altered nuclear morphology, that we would restore the flares, but you can see here that we haven't. That the double mutant is indistinguishable from Cdc5 no-flare on its own. So even though now these cells are small, they haven't grown, they still have the no-flare phenotype. So this tells us a couple of things. It tells us that the Cdc5 growth phenotype is not what's causing the altered nuclear morphology, and it tells us that Cdc5 is required to specify the nucleolus as the site of nuclear envelope expansion. And so now we can discount this model and start thinking about where to go next. And just to conclude, Cdc5 specifies the nucleolus as the site of nuclear envelope expansion, as I just said. And even though, unfortunately, we're not too much closer in working out the mechanism as to how Cdc5 is actually doing this, through the experiments I've been doing, we found actually two very interesting new phenotypes. And that is that Cdc5 appears to inhibit cell growth during a mitotic arrest, and also that SEC mutants display a really interesting bilobe nuclear phenotype. So the question is this bilobe forming to allow the maintenance of the nuclear cell volume ratio, and what the mechanism by which this bilobe forms. And with that, I'd like to thank everyone in the Cohen Fix Lab and the NIH for giving me a job.
Time for a couple questions. Here's one.
So, did you check the phenotype of this Cdc5 NF, like in other defects, like in mitotic acid or something else?
It has all those same defects, yeah. So its terminal phenotype is an arrest at telophase. We've tried all those are not the reason. If we take other mutations that affect mitotic exit, we don't see this phenotype. So we've discounted it working through MEN or FEAR or RDNA condensation. We've looked at many of its other mitotic phenotypes, and it doesn't appear to be acting through those.
Thank you.
Do you know how generalizable this phenomena are to other model systems?
Sorry, can you say that?
Do you know how generalizable this, what you saw is to other model systems?
Yeah, so
I'm
obviously...
known zebrafish.
Yeah, okay. Yeah, so obviously yeast do their nuclear division somewhat differently in terms of the nuclear envelope stays intact throughout the whole cell cycle. And so the nuclear envelope doesn't break down. You see that elongation, it stays intact, which is not what zebrafish do. The mammalian cells break down the nuclear envelope and then reform it once they've completed mitosis, right? So But nuclear envelope expansion is still important there to allow the decondensation, for example, the decondensation of chromosomes after the membrane reforms. So as a model in terms of, obviously mammalian cells don't make flares, they don't do the same thing, but nuclear envelope expansions are still important there, so this could still tell us something interesting.
They make projections though, right? Mammalian cells can make these projections from marine environments. Maybe you can repeat that.
Yeah, sorry, Mike was saying that they make projections. Yeah, but I don't think that's I don't know if that's relevant in terms of cell cycle and yeah.
I'm going to make this a bar question.
Sounds good.
So we'll move on.